All right, so I guess we'll go ahead and get started. Welcome everyone to the last Data Wednesdays of the semester. I hope everyone, at least in Rhode Island, is staying warm as the winter storm approaches. I am excited to introduce our speaker, Sriram Sankararaman, who is an assistant professor in the departments of computer science, human genetics, and computational medicine at UCLA. His research interests lie at the interface of computer science, statistics, and biology. His lab develops machine learning algorithms to analyze genomic data and clinical data with a broad goal of understanding the interplay between evolution, genomes, and traits. He received his undergraduate degree in computer science from the Indian Institute of Technology in Madras, a PhD in computer science from UC Berkeley, and he was a postdoctoral fellow at Harvard Medical School before joining UCLA. He is a recipient of an NSF Career Award and fellowships from Microsoft Research, the Sloan Foundation, the Okawa Foundation, and the Simons Institute. And I will let him take over and share his research with us.
Great. Thank you so much, Chelsea, for that introduction. Really excited to be presenting you all today virtually. And I'm completely flexible, so feel free to jump in if you have questions or put it in the chat. Although I can't see the chat from my screen, or we can put it to the end. So as Kelsey mentioned, my lab at UCLA is broadly in analyzing different types of genome data using from statistics and deep learning. And we use these tools and these analyses to elucidate different kinds of processes at the level of evolution. We try to understand the genetics and genomics complex traits, and we use it to inform clinical decision making. And while these themes might seem disparate, ultimately what interests me is the interplay between these different goals. So how evolutionary understanding can drive a better understanding of complex tree architecture, how we can leverage technologies and infrastructure built for complex trait analyses to better understand evolution.
I'm sorry to interrupt, Sriram. I think there's a little bit of an echo on your end. Do you have another mic or speaker going?
Sometimes it's the headphones.
Can you hear me?
Yes, that sounds a lot clearer, I think.
Okay, so I'll stick to this.
Thanks.
Okay, so so in today's talk, I'll mostly focus on complex traits. But before I jump in, I'll spend a few minutes talking about some of the work that we've done in understanding evolutionary processes. So we've been interested in this notion of ancestry. So it's a fundamental notion in understanding evolution. So where do we come from? Who are our ancestors? Who are we most related to? And I became interested in understanding ancestry in the context of searching for disease genes. So in the search for disease genes, genetic ancestry is a confounder, an important confounder. At the same time, genetic ancestry can be leveraged in interesting ways to design powerful ways of searching for disease genes. The problem, of course, is when you talk about genetic ancestry in the context of human populations fundamentally are admixed. So you have different populations splitting and then coming back together and mixing. The consequence of admixture is that when you look at ancestry, the ancestry is changing both across individuals as well as along the genomes of a given individual. And the final complication in all of this is we don't get to see this ancestry. It needs to be inferred from data. So my group has been working on statistical methods that can infer genetic ancestry and with particular applications to recently admixed populations like African Americans and Latino populations. And we've been using these methods, again, to design better gene mapping studies, but also to understand evolutionary processes. For example, to understand assortative mating patterns in the study that we looked at where we analyzed Mexican and Puerto Rican individuals. More recently, we've become interested in ancestry at a much deeper evolutionary timescale. So not just within the last hundreds of years in the Americas, but at the timescale of tens to hundreds of thousands of years. And a lot of these kinds of questions have been motivated by the field of ancient DNA, which is now giving us genome sequences from these archaic populations, particularly our closest evolutionary cousins like the Neanderthals and their sister group, the Denisovans. So by analyzing these archaic populations together with modern human genomes, what we've learned is this history of human populations, where it's not just one where human populations are splitting and diverging, but one where these populations come back together and introgress. And what we have now found is that there has been these major introgression events involving Neanderthals and the ancestors of present-day non-Africans, and Denisovans and the ancestors of present-day Oceanian populations. So as a result of these introgression events, present-day populations, again, carry or inherit introgressed archaic DNA. And there's been immense interest in being able to identify these positions where modern humans carry archaic DNA, which again leads us to statistical questions. So we've been interested in methods that can go from genome sequences to a labeling of these bits of archaic DNA. And by analyzing these bits of archaic DNA, we are building a picture of where these introgression events might have occurred. So in the context of the Neanderthals, we now know that almost all non-African populations today carry substantial proportions of Neanderthal ancestry. In the context of Denisovans, it's primarily centered in Oceanian populations, although there are populations on mainland Asia that also carry Denisovan ancestry. Beyond how archaic ancestry is shared across populations, by looking at these maps, we can also figure out how it's distributed along the genome. So we are able to identify positions in the genome which are either enriched or depleted in archaic DNA. And that is particularly interesting because it gives us clues into the phenotypic and fitness consequences of this archaic DNA. And finally, in the last couple of years, a major focus for my group has been the picture or the story of archaic ancestry in Africa. So Africa is particularly interesting because of the high amounts of genetic diversity in present-day populations, as well as the diversity in its fossil record. However, a lot of our understanding of archaic ancestry in Africa is still underdeveloped, largely because we lack the right kinds of ancient genomes from Africa. So in the last couple of years, we've developed these statistical methods which are reference-free, so which are able to identify archaic lineages without having access to these archaic genomes. And applying these methods to West African genomes that we then had access to, we identify about 8% of these genomes as tracing their ancestry to a distant population. And comparing these segments to present day individuals, as well as Neanderthals and Denisovans, we find that this population is not particularly closely related to any of these populations. And in fact, with additional modeling, what we find is that this population likely split from the ancestors of modern humans prior to the Neanderthal modern human split. So we call this a ghost archaic hominin simply because we don't have a good representative for these kinds of populations. So a lot of ongoing work in my lab is now focused on a better characterization of this ghost archaic population, both in terms of geography and in terms of their biological impact. And a major ongoing question is to understand the phenotypic and selective impacts of archaic introgressed material. But now for the rest of my talk, I'm going to focus on a slightly different theme that has emerged for us over the last few years, connected to the analysis of complex traits. So this has to do with this notion of biobank datasets. And many of you are familiar with this picture in different forms. It just shows the growth in sequencing or the decrease in the cost of sequencing over time. And so this was a paper from 5 years back, which showed how the number of genomes that are being sequenced, the amount of data that's being generated by sequencing, is far outstripping any other source that we know of. And I like this paper because what they showed, or what they claimed, was there is so much genomic data out there that it dwarfs every other data source. For example, data from astronomy. And so, in tongue-in-cheek, they claimed you need now a new adjective. It's no longer adequate to say genomic data is astronomical. You need a new adjective, which they call genomical, to describe genomic data. So we have, on the one hand, these large collections of genomic data sets. On the other hand, these data sets are being generated in contexts like the health system and clinic. And so this is what gives rise to these biobanks. So on the one hand, you have genetic information, genomic information, paired with very rich phenotypic and trait-related information. And we know of several examples of these kinds of data sets. The most prominent of this is the UK Biobank. So this is a data set of around 500,000 individuals in the UK. But there are many such efforts ongoing, both in the US and in many other countries. So this biobank data leads us to an interesting question, which is, what can we hope to learn about evolution and disease from these biobank scale data sets? So data sets that today already contain hundreds of thousands of genomes, thousands of traits, but in the conceivable future will carry millions of genomes. And for a lab like ours, related to the what can we learn is the how can we learn about evolution and disease. So specifically, what are the challenges and how can we overcome those challenges to learn about evolution and disease from these data sets? So the challenges are going to be statistical. So the data sets, these biobank data sets, are high dimensional, they're noisy. So you need to be extremely careful about the kinds of statistical methods that you can use on them to get valid inferences. The other challenge is computational. Clearly, there is a question of scale. So when you have these extremely large data sets, even the simplest analyses turn out to be quite impractical. And finally, there's the logistical challenge. So there is data sharing issues, there are issues of privacy. So how can we perform analyses while respecting notions of privacy that participants have? And so we've been interested in all of these issues. And today we're going to primarily focus on the first two challenges. So to make things concrete, I'll talk about two key inference problems that come up in the context of biobank scale data. So here is an example where we have genotype data. So we have SNPs collected on a few individuals paired with some trait phenotype on this same set of individuals. So given genotype and paired phenotype data, we'd like to be able to understand a few key questions. Firstly, given just the genotype, we'd like to understand what is the structure of the human populations that have contributed to these genotypes. So what were the relationships amongst the individuals who contribute to this data set? And as I mentioned in the beginning, this is of interest if we care about evolution. It is also of interest for the next question when we are trying to analyze the architecture of complex traits. So when we want to know what are the genes, how these genes come together to shape variation in the trait, that is a key question in and of itself. So first, I'm going to talk about the problem of inferring human population structure. So given genotype data, we'd like to say something about population relationships. And a widely used tool in population genetics is this tool called principal components analysis, PCA. So starting with genotype data, PCA converts this high dimensional data set. So this is a data set that is measured across millions of SNPs into a low dimensional representation that looks like this. So every individual here gets mapped onto, say, a 2D or a 3D representation, or a lower dimensional representation in general. And then we can visualize the relationships amongst these individuals. So this is a famous example of the application of PCA, where they applied this technique to individuals' genomes from Europe, and they essentially recapitulated the countries of origins of these individuals. So PCA is widely used. Now, what's the challenge? On the one hand, there is a computational challenge. So the computational algorithm that is used to compute PCA is something called the singular value decomposition, SVD. And if you have a data set with m SNPs and n individuals, the computational complexity is quadratic in the smaller of m and n. What does that mean? So it means if you have small numbers of individuals and many SNPs, or if you have a small number of SNPs and many individuals, you can compute PCA efficiently. However, for biobank scale data, so data sets like the UK Biobank with 500,000 individuals, tens of millions of SNPs, these kinds of methods, the simple PCA is infeasible. Of course, recently there's been a lot of work developing what are called randomized approximation algorithms, and I will talk about that in just a bit. In addition to the computational challenge, there is a statistical challenge. So what is the statistical challenge? PCA is a procedure. So if you give it generic data, it gives you an output. What is not clear is what are the assumptions under which PCA gives you the correct answer or the right answer. In other words, what is the statistical model that PCA is trying to solve? Here's an example where this kind of an issue matters. Let us say we have missing data, often genotype data in the context of these biobanks has missingness. We have to fill in this missing data. And how you fill in missing data can affect your inferences. So knowing what the statistical model is that is underlying PCA becomes crucial when you're trying to deal with issues like missing data. So how do we solve this problem? So the first thing we did is we tried to step back and ask the question, what is the statistical model that is underlying PCA? So this has been answered in a series of works in the statistics and the machine learning literature. And this goes by the name of probabilistic PCA. So the idea is you define a probabilistic model which gives you PCA as its solution. So here is a probabilistic model for PCA. So we are starting with n individuals who are measured on m SNPs. So this is genotype data on n individuals at m SNPs. So each of these genotypes are represented by this vector, xn. And now we're going to write down a statistical model that generates this data. So if you simulate data under this model, you will get our genotypes. So the statistical model for probabilistic PCA says the following. For every individual, we're going to generate a random low dimensional vector. So it's a vector of dimension k. Given this random low dimensional vector, which we don't know, it's a hidden quantity, we're going to generate our data by taking this low dimensional vector, transforming it by a linear matrix to which we add noise. So we do this independently for every individual, and that gives us our data set. So what's special about this model is you have a statistical model. So now you can infer the parameters of this model. So the parameters of this model are this matrix W and these latent low dimensional random vectors, Z for every individual. And what is interesting about this model is if you compute the maximum likelihood parameter estimates, these maximum likelihood parameter estimates correspond to the principal components and the principal component scores. In other words, this is equivalent to essentially running PCA. So now why is this useful? So estimating the principal components is now a problem of computing maximum likelihood estimates in a statistical model. And this is a special kind of statistical model because it is a model where there are hidden variables. And we know ways of performing maximum likelihood estimation in a model with hidden variables. And the way we do this is using something called an EM algorithm. So the EM algorithm is a classical algorithm which allows us to compute maximum likelihood estimates in models with hidden variables. And what it does is it's an iterative process. We have a model with two unknowns, the principal components and their scores. We assume one of these is known and estimate the other, and then we repeat that process. So we assume that the principal components are known and we estimate the scores. Having gotten the scores, we recompute the principal components and we repeat. And the reason why this is useful in the context of large data sets is that each of these steps of the EAM algorithm has a runtime that looks like this. So K here is the number of principal components that we are trying to compute. And for most applications, K is small. Often we are interested in the top 10, top 20 principal components. So the runtime of each step is a linear function of both the number of SNPs and the number of individuals. As a result, it is an extremely efficient algorithm. Turns out you can actually reduce the runtime even further, and that's due to some additional structure in the problem. So for those of you who are interested, the reason why this runtime has a form that looks like this is because the genotype matrix, so the matrix X, is a matrix of zeros, ones, and twos. And every step in the EM algorithm simply involves repeated multiplications of these genotype matrices. And so by exploiting this structure, it turns out we can further reduce the runtime of the EM algorithm. So we implemented this in a package called ProPCA, and we benchmarked ProPCA on a number of different data sets. So the first one was an accuracy benchmark, where we have populations with different FSTs, different levels of divergence. We're trying to estimate the top 5 and the top 10 principal components. And this is a metric which tells us how close is the ProPCA principal component compared to the true principal component. Essentially, we see that it's extremely accurate. Further, we also can benchmark its runtime, and we find that the ProPCA algorithm runs as a linear algorithm. So as you increase the number of individuals, it scales in a linear manner. So this second plot here compares different algorithms. So this is the algorithm that you would run if you were doing a singular value decomposition. We see that it doesn't scale beyond a few tens of thousands of individuals. On the other hand, there is ProPCA that scales linearly. And we've also plotted a few other algorithms that are all based on what we call randomized projection. So these are all different randomized algorithms which also scale linearly. However, ProPCA is still the most efficient amongst all of these algorithms. So now we applied this to the UK Biobank. It turns out we can compute the top 5 PCs on the full UK Biobank in about 30 minutes on standard compute infrastructure. And so if you look at the first and the second PCs on the UK Biobank, for the full UK Biobank dataset, we recapture what we know about human population structure, where the first principal component essentially separates individuals of African ancestry from individuals of non-African ancestry. PC2 then separates European individuals from the rest. Because we also have a large representation of individuals from the UK, we don't only have to look at structure that's intercontinental, we can look at structure within the UK. And so this is the application of ProPCA to individuals in the UK Biobank, where we have now plotted them along with the geographic coordinates. And what we see here is the first PC score for all of these individuals. So it tells us where each individual would fall along the first principal component. And what we see is that the first PC is essentially capturing a north to south climb within the UK. Similarly, as we go further down the PCs, we get additional insights into fine scale structure within the UK. For example, the second principal component seems to be a PC that's separating out individuals from Wales and so on. So now that we have computed these principal components, we have insights into this fine scale population structure. What can we do with this? What additional things can we learn? So one interesting idea is that these principal components tell us the general patterns of relationship across the genomes of these individuals. So now we can ask, are there regions of the genome that are not well represented by this general geographic pattern? And the reason that could be interesting is some of these regions could be regions that are evolving much faster than what is represented by the average relationship across the genome. So that would be signals of recent positive selection within the UK. So we applied this. So we have a statistical test which goes across the genome and searches for regions or SNPs that are not well represented by the principal components. And so what you see here is a Manhattan plot where we have the p-value. So points that are high up are SNPs, which are statistically significantly not well represented by the genome-wide principal components. It turns out that many of these are loci that have been found in other scans, in other studies. So these are loci that have strong evidence for being under positive selection. So we find examples like the lactase gene, the TLR gene, which is well known to be important for immune-related function, HLA, and the FUT2 gene, which has been shown to be important for blood pressure-related traits. But in addition, we also recover a handful of novel loci. So these are, again, loci which are likely to be under recent positive selection. This includes the HERK2 gene, which has been shown to have an important role in pigmentation, and the RGRIP1L gene, which has been shown to be important for neurological conditions. So by combining these large data sets with statistical methods that can be applied at scale, we are able to detect recent positive selection within the human genome. So now I'll switch to the second key inference problem in the context of biobank scale data, which is learning about the genetic architecture of complex traits. So now we are pairing genotype data with trait information. And by genetic architecture, what we mean is inferring the function that relates genotype and environment to the trait. So clearly, this is a complicated problem. So let's again have a concrete example of what genetic architecture would mean. So one key quantity in understanding genetic architecture is this notion of heritability. So heritability is this notion of what proportion of the phenotype can be explained by genetics. There are several notions of heritability. So the one we will focus on is something called narrow sense heritability. So narrow sense heritability asks what proportion of variation in the phenotype can be explained as a linear function of genetics. So to estimate heritability, we need, again, a model. So we have phenotype data, we have a genotype. So we model the phenotype as a linear combination of the genotype plus environment. So the linear combination is represented by effect sizes for these SNPs. In principle, you can now fit a linear model given a phenotype and genotype data. The problem, of course, is we have way more SNPs than individuals in a given study. So you cannot directly fit a linear model. You need to put some assumptions on the effect sizes of these SNPs. So one common set of assumptions is to assume that the effect size at each SNP comes from some distribution where the variance of this distribution is related to the genetic variance component. Similarly, the environment is assumed to come from another distribution whose variance corresponds to the environmental variance component. So now we have a model which has a genetic and an environmental variance component. The heritability of this trait, the narrow sense heritability, is a function of the genetic and environmental variance components. So this class of models are called variance components models. What we've described is just a basic version of this variance component model. So in the basic version, every SNP, no matter where it lies in the genome, is drawn from the same underlying distribution. Now, of course, this is a simplistic notion, and there is plenty of evidence that suggests that SNPs in different parts of the genome might have different effects. For example, if a SNP is in a gene, it might come from a different distribution compared to a SNP that lies outside a gene. So now this variance components model has been modified, which allows for different effect sizes depending on where a SNP lies in the genome. Similarly, even if we only care about estimating heritability genome-wide, there's a recent work that shows that the effect sizes of SNPs can vary as a function of population genetic features like the minor allele frequency of a SNP. For example, if a SNP has low minor allele frequency, it might have a different effect size distribution compared to if it has high minor allele frequencies. So all of these can be handled by extensions of these variance components models. So broadly speaking, Our goal is to fit a variance component model where there's a phenotype. A SNP is assigned to one of k possible genetic components. Each of these genetic components has an effect size, and all SNPs that lie within a genetic component, their effect sizes come from the same distribution. So having this model, our goal is to estimate all of the genetic variance components. In this case, there are k of them, and the environmental variance component. So now we again have a statistical model which has parameters, and we can estimate such a statistical model by maximizing the likelihood of this model. The challenge here is maximizing the likelihood is computationally expensive. So methods for maximizing likelihood typically scale cubically in the sample size. And there's been plenty of work, algorithmic work, that tries to make it more scalable. Nevertheless, it's challenging to apply these methods to biobank scale datasets. So we started thinking about this problem, and to try to make progress on this, we formulated an alternate way of performing estimation. So instead of doing maximum likelihood, we looked at an alternate estimator, which is what we call method of moments estimators. So the idea of method of moments estimators is very simple. The goal is to select the parameters of this model so that the moments, the population moments, match the sample moments. So in the context of the model that we just described, we're going to look at the second moment, the covariance of the phenotype. So the covariance of the phenotype can be written as a linear combination of the variance components weighted by these matrices, which we call the genetic relationship matrix. So the genetic relationship matrix, or the GRM, is a matrix for every pair of individuals in the dataset that measures how similar the pair of individuals are at the SNPs that correspond to that specific variance component. So now, the method of moments requires us to pick values of these variance components such that the population covariance matrix matches the sample covariance matrix, which we estimate from our data. So the nice thing about these method of moments estimators is we can actually compute the method of moments solution in closed form. So they're easy to compute, almost. The challenge is you still have to compute these genetic relationship matrices, these K matrices. And as I said, this is a matrix for every pair of individuals. So even computing this matrix scales quadratically in the sample size. So again, for biobank scale data, this kind of an approach doesn't work. So the final insight that allows us to come up with a scalable method is that we can actually compute these method of moments estimators without ever explicitly forming this genetic relationship matrix. And the insight is to compute this method of moments, we only need not the GRM, so we don't need these K matrices, we only need the trace of the GRM or trace of products of the GRM. And the reason this is useful is there's prior work from the randomized linear algebra community that tells us how we can estimate this trace by a randomized estimator. So that's what we have here, where we have replaced the trace or approximated it by a randomized estimator. And what that involves is taking this matrix and multiplying it by some number of random vectors, where each random vector is a vector with mean zero and identity covariance. The reason why this is a computation that's useful is because now each of these involves simply computing a random matrix vector multiplication, where the matrix is the genotype matrix and the vector is the random vector that we have chosen. So all we require to do is multiply the genotype matrix with some selected number of random vectors, which we call B. So the B is a parameter of this algorithm, and the efficiency depends on the value of B. So if you have chosen the number of random vectors, then the computation of this algorithm scales as linear in the number of SNPs and the number of individuals. Again, the denominator here, the log, is simply because a genotype matrix can be multiplied with these random vectors much more efficiently than a typical matrix. Now, what was surprising to us is that this method, this randomized Huysman-Elston regression method, gives us accurate estimates of heritability for values of b as small as 10. So in this computational complexity result, b is small, which means essentially this algorithm scales linearly with the number of SNPs and the number of individuals. So we ran some simulations to validate our method. And so these are simulations under a variety of different genetic architectures. So we have simulations with causal SNPs chosen randomly along the genome or enriched for low frequency or common variants. We also explored different ways of coupling the effect sizes of the SNPs to population genetic quantities like the minor allele frequency, LD, and so forth. And what we find is across these simulations, the randomized Huysman-Elston regression method performs about as accurately as maximum likelihood. So maximum likelihood is the gold standard here. Both methods are relatively unbiased. The randomized Huysman-Elston regression has slightly larger standard errors, about 10% bigger than maximum likelihood. So the simulations that we just talked about were all done on small numbers of individuals, 10,000 individuals. And the reason for this is simply because maximum likelihood cannot scale to these larger sample sizes. So we were also interested in whether this method can actually scale to large sample sizes. And so here we assessed its accuracy in these large sample settings. To compare its accuracy in large sample settings, we compared it to methods that can scale. So these are methods which are based on summary level data. So the most famous of this is something called LD score regression, which can estimate quantities like heritability without access to all of the individuals, instead working just off summary data. So what we found is all of these summary level methods tend to have relatively large biases across these simulations, whereas our proposed randomized Huysman-Elston regression is relatively unbiased. And the reason for this is these summary level methods are making different assumptions. So when these assumptions are violated, that leads to bias. Whereas the randomized Huysman-Elston regression method, because it is able to adapt to the assumptions underlying these simulations, is relatively unbiased. So we also validated its scalability. So as we increase the sample size all the way up to 300,000 individuals, which is the number of white British individuals in the UK Biobank, the randomized Huysman-Elston method can actually scale, running in a matter of a few hours, whereas the methods that try to compute maximum likelihood solutions do not scale. In fact, we've run it for data sets as big as 1 million individuals and 1 million SNPs. And we can also scale to large numbers of genetic components. So for example, we can have hundreds of components and we can estimate heritability and how heritability is distributed across these hundreds of components. So those were all simulations. Now we applied these different methods to about 22 complex traits and diseases measured in the UK Biobank. And we estimated the genome-wide heritability from different methods. So what we found here is generally the randomized Huysman-Elston regression method that we proposed estimates lower heritability relative to the other methods, which are all based on summary statistics. So we think that this is because of additional biases that are being incorporated into these summary statistic-based methods. Because the method can scale, we could actually apply it not just to common SNPs, we could also apply it to low frequency variants. So in this experiment, we looked at array data, which primarily has SNPs with minor allele frequencies greater than 1%, imputed data, restricted to, again, common SNPs with allele frequencies greater than 1%, and imputed data with allele frequencies going down to 0.1%. And what we find is the heritability increases as we include the low frequency SNPs. The proportion of increase depends on the specific traits, but on average across these traits, it increases by about 11%. So what this tells us is as we are able to sequence lower frequency variants, we will recover more and more of the trait heritability. As I said, the randomized Haseman-Elston regression method allows us to also partition heritability across the genome. So in this example, we examined how heritability for a trait varies as a function of the minor allele frequency and the LD patterns that are local to a particular variant. So we divided up the genome according to the minor allele frequency and the LD patterns at a SNP. And in this case, we plot the allelic heritability for height, which was one of the traits we looked at. And what we see here is as the minor allele frequency decreases, the allelic heritability of height increases. So this is consistent with the action of negative selection on height. So this has been documented previously, but we are the first to document it at these minor allele frequency ranges. Further, we also document a novel relationship where we find that the allelic heritability increases as a function of LD. So as you move towards lower LD SNPs, allelic frequency, allelic heritability increases. Again, this is consistent with negative selection, and there's ongoing work in our lab to figure out what this might tell us about the modes of selection. We can also partition heritability not just by population genetic annotations like minor allele frequency, LD, but by also functional genomic annotations. For example, annotations that come from epigenomic studies and regulatory genomic studies. So in this example, we have partitioned the heritability for 5 representative traits in the UK Biobank. And what we plot here is the enrichment in heritability across these annotations. So an enrichment of 1 says in that specific annotation, there is no enrichment of heritability. So it's at about the genome-wide average, whereas enrichment values greater than 1 tell us that there is substantially more heritability in that annotation than you would expect. As a general rule, we find that for all traits, there is enrichment of heritability in regions that are conserved across species. However, there are also trait-specific enrichments that we observe. For example, for autoimmune traits, we find that enhancer regions tend to have enriched heritabilities. So putting all of this together, by interrogating these large data sets across these diverse traits using these scalable methods, we are able to gain some insights into genome-wide heritability, as well as how heritability is distributed across the genome. One downside to all of this is the heritability that we think about accounts mainly for linear effects. So it's talking about what is the linear effect of a SNP on a trait. Clearly, that may not capture all of the genetic architecture. So what about nonlinear effects? Further, in a biobank like the UK Biobank, we have access to a large number of traits. So this gives us the opportunity of trying to understand how heritability is not just shared across the genome, how it might be shared across traits. So this leads us to the question of, can we estimate these effects in large biobanks? So we described a very specific model that can estimate narrow sense heritability, so this linear function on biobanks. Can we estimate nonlinear effects? Can we estimate heritability shared across traits in these large biobanks? So what is interesting, and I won't spend too much time on this, is the fact that these same ideas are extremely general. So they give us methods that can estimate nonlinear effects, heritability that is unique and shared across traits, all in the context of these large data sets. So here is one such example where we extended this method to estimate dominance effects. So we are looking at a trait and we are trying to ask how much of the variation in the trait comes from additive effects versus dominance effects. And again, this can be done efficiently using the same randomized method of moments idea. And applying this method to about 50 traits, quantitative traits in the UK Biobank, what we observe is across these 50 traits, there is substantial evidence for additive effects, additive heritability, whereas dominance heritability seems to be substantially smaller. So on the whole, dominance heritability seems to be about 2 orders of magnitude smaller than additive heritability. And in fact, when we do a formal statistical test, we do not find any evidence for a trait which has non-zero dominance heritability. What is also interesting here is because of the large sample sizes, we can also put very strong bounds on the largest possible dominance effect. And our analyses suggest that no trait amongst the 50 that we analyzed has dominance heritability greater than 1%. So compared to the typical additive heritability for traits being of the order of 20%. The other important aspect of these biobanks, as I mentioned, is we have access to many different measurements. And this gives us the opportunity of asking whether there might be interactions between the trait that we care about and measures of the environment. For example, if we are interested in lung capacity and we have access to smoking, we might be interested in asking, is there interaction between smoking and genetics in its influence on lung capacity? So in more recent work, we have extended these ideas to estimate these gene environment interactions on biobank scale data. And again, I won't spend too much time on the validation results, but just talk about analyses of data. So in this analysis, we have taken our environment to be smoking, and we tested for G by E, gene environment interactions, for many different traits. And what we find is evidence for G by E for a number of different traits and substantial contributions in terms of G by E. For example, we find that smoking and genetics together, they affect the heritability substantially for traits like BMI, cholesterol, lung capacity, and urea. In fact, in urea, about 30% of the additive heritability comes from gene environment interactions. On average, across these traits, it's about 5%. So there is quite a bit of substantial gene environment interactions contributing to variation in many of these traits. The other aspect that we were particularly interested in is trying to understand how genetic effects might be shared across traits. So this is a concept that is called genetic correlation. So genetic correlation measures what fraction of the correlation in a trait is driven by correlation of genetic effects. So here's an example where we have two traits, height and BMI. Height and BMI are correlated at the level of phenotypes. So if you measure the correlation between BMI and height, you might get a negative correlation. But then if you knew what were the causal variants for height and BMI, you might measure the effect sizes of these causal variants on height and BMI. And then you can ask, what is the correlation of the effect sizes of the causal variants for these two traits? And in this case, we might conclude that the genetic effects at the causal variants for these two traits are positively correlated, while the traits themselves are negatively correlated phenotypically. So in this case, the genetic correlation would be negative for BMI and height. So we were interested in understanding genetic correlation and estimating it again from biobank scale data. So we could now extend our variance component model to account for two traits simultaneously. So now we have two traits. We have the effects of genotypes on each of these two traits. And when we look at the effect sizes of the genotypes on each of these two traits, we model them by a bivariate distribution, where there is a coupling between the effect sizes of the two traits. So this quantity, this gamma g, measures the coupling of the effect sizes. So a gamma g of 0 means there is no correlation in the effect sizes. Positive means there's positive correlation, negative means there is negative correlation. And now the goal is to be able to estimate these coupling parameters, which are related to the genetic correlation, in addition to all of the other parameters related to heritability. Again, it turns out this randomized method of moments can also be used to estimate genetic correlation at scale. So here are some experiments where we looked at the performance in terms of accuracy of this method, which we call SCORE. So SCORE has standard errors, which are substantially smaller than the other scalable methods. So for example, LD score regression can estimate genetic correlation from summary level data. HDL is another recent method for estimating it from summary level data. For example, when we compare to LD score regression, the reduction in the standard error roughly translates into a threefold increase in sample size. Relative to HDL, it translates into an 85% increase in sample size. And of course, compared to maximum likelihood methods, score is extremely computationally efficient. So it's orders of magnitude faster than methods that compute maximum likelihood. So we then applied score to 28 different traits in the UK Biobank. So for every pair of traits, we computed its genetic correlation, and we compared it to genetic correlation estimates from LD score regression. So in the upper diagonal, we have estimates from LD score regression. In the lower diagonal, we have the estimates from score. And the stars are traits that are found to be significant for genetic correlation by score, but not by LD score regression. So we find that score detects an additional 58 pairs of traits as having significant genetic correlation. And that's again because it has much higher precision. Again, because of the richness of the data, there are a large number of traits that we can analyze. For example, the UK Biobank has, in addition to complex traits, it has a number of blood biomarkers. And so we were lucky enough to have a collaborator who pointed us to this data and said that there were interesting questions, interesting biology to be discovered by running our method, connecting complex traits with blood biomarkers. And when we did this, we find that there is a significant genetic correlation between heart-related diseases, so coronary artery diseases like heart attack and angina, and these liver enzymes that are measured in the blood, for example, alanine aminotransferase. And this is a novel genetic correlation. And one of the reasons why this is interesting for our collaborators is because this points to a link between liver function and heart diseases. So there is interesting follow-up work that we're doing to understand the basic biology underlying this genetic correlation. So just to summarize, hopefully what I've been able to show you here is that there are major promises in terms of the kinds of things we can learn from these biobank scale data sets, but there are accompanying challenging problems that need to be solved. Many of them are of the flavor of challenging statistical inference. So I gave two kind of examples. The first one was about population structure inference using principal components analysis. And the way we approached this was the view of statistical modeling. So thinking of PCA as a statistical latent variable model. And what was interesting about it is it tells us what are the assumptions underlying PCA, but it also gives us efficient inference algorithms. And using this efficient inference, we are able to compute fine scale structure in the UK Biobank, which in turn provides us insights into targets of recent positive selection. In terms of ongoing work, PCA is just one way of quantifying or visualizing population structure. There's been a huge amount of work in what are called admixture models or structure models. And it turns out that a lot of the insights that we have for PCA can also be leveraged to perform structure inference at the scale of biobanks. So I'm happy to talk about that if anybody's interested. The second class of analyses I talked about is variance components analyses. And so one high-level way of thinking of this is we have maximum likelihood methods, which are extremely accurate, but are computationally intensive. There are summary statistic methods, which are not so accurate, but are computationally efficient. And we can think of these randomized method of moments that we discussed as trading off accuracy and runtime. So it's about as accurate as maximum likelihood estimation while being extremely efficient to run. And we presented these randomized method of moments algorithms in the context of estimating heritability and partitioning heritability, in terms of estimating dominance and gene environment interactions, and estimating genetic correlation. One thing that's quite surprising and interesting for us is the generality of this kind of analysis. And so we think that this can be extended to a large number of settings. And in terms of the applications, we use this to inspect the action of negative selection on complex traits. We use this to understand how much additive versus dominance heritability exists in complex traits. We find that there is substantial additive but negligible dominance heritability. Further, we are able to now see evidence for substantial gene environment contributions for specific combinations of genetics and environmental variables. As I said, there's a lot of interesting possibilities and directions for future work here. So some of the things we're thinking about are biobank scale association testing and prediction. So heritability only gives you a genomic view of what might be important, where genetic signal lies. Often we care about localizing that signal. So that's where testing, association testing comes into the picture. Again, a related quantity is prediction. So how do we actually make predictions on new genomes given the biobank scale data sets that we've collected? It turns out a lot of these insights can be leveraged for those kinds of analyses as well. Another area of a lot of importance is performing multi-ethnic analyses in admixed populations. Again, this is another place where having diverse populations in these biobank data sets is extremely important. So how do we actually have methods that can work on these settings is going to be a major challenge for the community. And finally, combining different data modalities. So everything we've talked about here was essentially for a simple kind of scalar way of measuring phenotypes. But what's so exciting about these biobanks is that's not the only kind of data you have. You have molecular data like gene expression. We have data like imaging. We have sometimes time series data. And so putting all of these together, often as intermediate phenotypes to better understand disease risk, is going to be a major possibility in terms of opportunities and also interesting challenges. All right, so with that, I'll conclude, and I'd like to thank all my students, my wonderful students who did all the work. So a lot of the work on heritability estimation, dominance, gene environment interactions was led by this very talented student, Ali. Ariel worked on the genetic correlation estimates, and the PCA work was led by Alec and Aman. And I'd like to thank funding sources. And I just want to end by saying this was us hiking into the Santa Monica Hills about a year back, but it probably seems like sometime in the distant past. So right now, the only way we're going to see each other is over Zoom. But hopefully that'll change relatively soon. Thank you.
Thank you for sharing your work with us. Does anyone have any questions?
So how long did it take to run the probabilistic PCA on the UK biobank data?
It probably depends on how many PCs you want to compute. So we just computed the top 10 PCs, and it was on array data, so about a couple of 100,000 SNPs. And that took us about 2 hours.
Okay. Thank you.
I have a question. I'm not sure if it's a good one because I'm a molecular biologist. So, but I'll hazard it. One thing I've often wondered for diseases that have both genetic and environmental components. So if you have maybe, I don't know, 10 to 20 genes that are associated as risk factors for a disease, and you have maybe 4 to 6 environmental factors that are risk factors for the disease. Is it possible to use these kind of analyses to figure out which of the genes are subject to the gene times environment interactions to get a sense of how the environment is feeding into the genetics of the disease? Does that make sense?
Yeah. So is the question to identify whether on aggregate there is an effect of any of these environments, or the question be to identify specific environmental effects.
The question is, say you've already identified specific environmental effects and you want to know which genes are mediating those effects.
I see. Yeah, I think we haven't tried it. So generally what happens to these methods is as you try to get smaller in terms of the resolution, these methods generally have less power. So in other words, if I was asking the question, does this chromosome mediate an effect? Or does this pathway mediate an effect? Versus does a specific gene mediate an effect? The last analysis is going to be the hardest, the least powered compared to the first one. And that's because there's going to be LD between the variants that lie within a gene and nearby variants, which makes it hard to figure out, is it exactly this gene or could it be some position that's nearby that might actually be mediating this effect? But this was all done. I mean, a lot of this was observed in much smaller data sets. And it's quite possible that in these larger data sets, you might actually have the resolution. And so a lot of our current work is at trying to figure out how small can we go to be able to narrow this signal. So I guess the short answer is, I think it's possible, but we haven't shown with certainty that it should be.
That's really interesting. Thank you.
Any other questions? All right, well, thank you so much for joining us today, Sriram, and thanks everyone else for coming, and everybody have a great holiday.
Thank you.